Hey, welcome to my channel Biofreak. Let's talk about amino acids. Amino acids are the building blocks for proteins. Immunoglobulins or antibodies that fight against infections in our bodies are proteins. Actin and myosin in our muscles that enable muscle contraction and body movement are proteins too. Several hormones, enzymes, keratin in your hair and nails, hemoglobin that carries oxygen in your body are all proteins. In fact, the collagen is the most abundant extracellular matrix protein in mammals. So you can see how widely proteins are distributed in our body, and life is just impossible without proteins. The monomer amino acid will form the polymer of protein, and hence it becomes very important to talk about this building block, amino acid. Roughly 500 amino acids have been identified, but out of them in this tutorial video, we will be talking about major 20 amino acids that constitute mammalian proteins. Each amino acid consists of one carboxylic acid group, which is shown over here, which is also acidic in nature as per the name. There is amino group that is basic in nature. There is one R group or a side chain and one hydrogen atom that is attached to this carbon. Now this carbon is referred to as alpha carbon. Now in all amino acid, everything remains same except for this R group, and R group varies in each amino acid. You can actually distinguish or categorize this amino acid like non-polar, polar, acidic, basic or aromatic based on the type of R chain you see. So let's begin first with the non-polar amino acid. There are 9 amino acids that are non-polar, and non-polar meaning they act like oil droplet or lipids. So their R chain is hydrophobic in nature. So we know that proteins will fold, right? They fold in 3-dimensional shape. So when this amino acid, the non-polar amino acid fold, the R chains are buried in the interior of the protein so that they don't come into direct contact with the aqueous solution because they are hydrophobic in nature. Now the first 3 amino acids are displayed on the screen, and the first one that is displayed over here is glycine. Glycine, remember guys, that's the simplest amino acids out of all because the R chain over here is nothing else but single hydrogen atom. Now you can see this alpha carbon is bearing 2 hydrogen atom, one amino and carboxylic acid group as usual, but this 2 hydrogen atoms will make this amino acid optically inactive and the carbon is achiral. So what happens is that the mirror image becomes superimposable. So achiral. Rest all amino acids are chiral in nature, they are optically active too. Exception, the only exception over here is glycine, which is very important to be remembered, as these are the questions which are asked by the examiner in competitive exams. So guys, please make note of this. Moving further to this, isoleucine. Isoleucine is actually the isomer of another non-polar amino acid, that is leucine, which we'll be looking into next slide. Methionine is another important amino acid. Why am I saying important? It has this sulfur containing R group. Ethyl, methyl, thiol, ether. Okay, so this is methionine. Now as we all know, in the translation process, if you know the central dogma, replication, transcription and translation, right? So in translation process, you have that start codon AUG that will code for this particular amino acid, that is methionine, without which the translation does not initiate. So you can see it is also important one. Moving further, there is alanine, valine, leucine. As I said, leucine, and it has its own isomer, that is isoleucine. Alanine, valine, leucine, they all have this aliphatic R group. Now remember guys, when we talk about the non-polar amino acid, the R chain can be either aliphatic or aromatic. So let's look at 2 of the aromatic amino acids. So you have tryptophan and phenylalanine under non-polar amino acid category, and this tryptophan has heterocyclic indole structure. So this is the indole structure over here. Okay, it has this heterocyclic structure. This is aromatic amino acid. Phenylalanine again has benzene ring, aromatic R group. Just remember this too, again very important. Talking about another exceptional and very important amino acid, that is proline. Proline is the only one amino acid that has secondary amino group. Remember guys, this is the only amino acid that has secondary amino group, and it is frequently referred to as amino acid. Okay, and this also has this 5-membered ring structure. So this is what? This is proline. So this was all about the 9 amino acids which are non-polar. Let's move on to the polar amino acids. Now the polar amino acids are hydrophilic in nature due to the R chain, and the first two are serine and threonine. As we all know, this is which group? Hydroxyl group. Threonine and serine are the 2 amino acids which are hydrophilic in nature. They are polar. Cysteine is another amino acid in a polar category that is sulfur containing, that has sulfur in it. And you can see this cysteine in insulin hormone. It forms what? It forms disulfide bonds, right? So it is again very important. 2 sulfur containing amino acids that we had learned. One is from non-polar category, that is methionine, and another one is cysteine under polar category. Talking about tyrosine. Tyrosine is aromatic amino acid under polar group, and it has this phenol group. This is phenol, right? Moving further, there is asparagine and glutamine. They both have amide group, CONH2. So this, what you can see over here, this is amide group. This is also amide group. So asparagine and glutamine, they both have amide group and they are polar in nature. Talking about acidic side chains, aspartic acid and glutamic acids are the examples, okay, for acidic amino acids, and they have acidic side chain, as in, you remember that initially I said that COOH, carboxylic acid, is acidic in nature, and this amino acids have extra carboxylic acid group attached to their R group. This adds up to the acidity. Same thing applies over here. There is another carboxylic acid group in addition to this particular carboxylic group. So you can see that there is acidity that is increased. Talking about the charge, okay, this can be easily deprotonated. Okay, whenever we talk about any acidic group, even here, okay, acidic group can be deprotonated. Okay, they can lose this 1 proton and they can get net minus charge. So usually you can say that they are negatively charged amino acids. Moving further to the basic amino acids. Basic amino acids, they have extra amino group over here. You know, lysine and arginine, they have extra amino group. Charge is positive. Why? Because they can be easily protonated. Since they are basic in nature, they can be protonated. They can take up extra proton and get net positive charge. So lysine and arginine are the 2 amino acids which are, you know, found in histone proteins. So the DNA that is negatively charged will wrap around this positively charged histone protein, and histone protein is rich in lysine and arginine. That's the link here. Histidine is another one that has 5-membered imidazole heterocyclic structure. Okay, so this is heterocyclic structure. This is another, you know, exception over here, or something different about this particular amino acid which can be focused on in examination. So this was the chart about whatever we have discussed till now, all the groups and whatever was very important. You guys can take a screenshot of this. Remember this part, very important part over here. So glycine is the only amino acid that is optically inactive. Methionine or cysteine, they are the sulfur containing amino acids. Phenylalanine, tyrosine and tryptophan are aromatic amino acid. The proline is the only amino acid that has secondary amino group. Serine and threonine, they have hydroxyl group, OH group. Asparagine and glutamine, they have this amide group. Lysine, arginine, histidine, they are basic in nature. They are positively charged. Acidic and negatively charged proteins are aspartic acid or glutamic acid. They are also referred to as glutamate and aspartate. So do not confuse with asparagine and glutamine with aspartate and glutamate. Just, you know, make note of that as well. Many people get confused between these two. Moving further, there is another way of classifying amino acid based on whether they are required by the body or whether body can synthesize them. You do not require. So, you know, if something is synthesized by your body itself, okay, and you do not take it via your diet or something, then it is not needed by your body, right? So it is referred to as non-essential amino acid. So this is the category of non-essential amino acid. On the other hand, okay, something that is not synthesized synthesized by your body and you have to obtain it from your nutrition or diet, that means it is essential for your body. So this is the least of essential amino acids. So that's how you can classify them, okay, on the basis of whether they are required by your body or your body can synthesize it. You do not require them from the external source. Moving further, this is kind of a property of amino acids. So whenever we talk about amino acids, okay, you won't find them in a particular, let's say, cationic form or an anionic form or zwitterionic form. Okay, they will be in the equilibrium. Okay, and it all depends on the pH. So let's say in a particular medium, okay, where the pH is very low and you have this amino acid, you will find them in the cationic form. Now why am I saying you will find them in a cationic form? Because you know that there is one amino group over here and that is basic in nature, and as I said, any amino group, it can be easily protonated. It can gain one H plus ion and it can get this net positive charge over here. So this happens at lower pH, okay, wherein the H plus concentration in the medium will be high. On the other hand, the exactly opposite situation wherein there is high pH, okay, and there is hydroxide ion, okay, the OH minus ions, okay, in the medium are higher. So this hydroxide ions can deprotonate any acidic group, and acidic group over here is carboxylic acid group. So you will get negative charge on COO minus, okay, the COH protonates, and you get this negative charge. So what happens here is that there is an ion formation, whereas at the physiological pH, okay, what happens is that there is zwitter ion formation, wherein a particular molecule will have both positive and negative charge, okay, and the net charge is 0. This happens at physiological pH. In our bodies, the 7 to 7.4 is the physiological pH, wherein you maintain this, your body maintains this pH very precisely, right? Or else it will lead to various disorders. So at this particular pH, what happens is that the balance is maintained and you will find these zwitter ions. Now what happens is that the proton from here, okay, the COOH will deprotonate and this one NH2 will get protonated. So this H plus will move on to this particular basic group, that is amino group. So that's how you get a zwitter ion. This is another important property of any amino acid. Okay, you can they are amphoteric in nature. They can act as acids and bases both. So in the beginning, I said that the monomer is amino acid, the polymer that is formed is protein, right? So here we are talking about the ligating factor. The monomers are to be ligated together. They should be linked together by something to form the polymer of protein, right? And this is done by the peptide linkages or peptide bonds. Now peptide bond formation is the condensation reaction over here between one amino acid and the other amino acid. So which group over here is participating? This is the amino group and this is carboxylic acid group. So OH from the carboxylic acid and one H from the NH2 amino group, they combine to form water. The water molecule is lost and then you get this peptide linkage in between. So that's how the peptide links are formed, and this continues on and you will get a long chain polymer and you will finally get protein. Now this is not the end of the process. There is also protein folding, and you know there are a lot many modifications that are done in our body to manufacture proteins ultimately, but this is the basic bond between the two amino acid residues that will form proteins. So that was it guys. I hope you liked this video and it was helpful. Please feel free to ask me any queries or doubts. Subscribe my channel for more such tutorial videos. Thank you.